Okay.
I have a question. The termination sequence, there's 3 up there.
Yeah.
Will there technically be the same amount on the RNA, or will there be fewer? I mean, I know you only do.
Oh, it's going to vary because the thing with the termination sequence, most genes have multiple termination sequences. They tell the polymerase it's time to fall off. Problem is the polymerase doesn't listen very well. So here let's suppose you're a termination sequence. I'm a polymerase and I'm chugging along and making some RNAs and stuff, and I go past you and then I continue on. Sometimes I'll get to you, oh, I'm supposed to drop off, bye, I'm out here. You know? But other times, I may read right past you. I might ignore you're there. Who cares about you, man? You know, and read right past. And I would continue to do that, except she's a second termination sequence. If you don't get me off, she will. Maybe. But I'm going to read through you two. And Sandy here is a third termination sequence, and I finally listen to her, and bye-bye. Now, that read-through is random. Sometimes I might stop here at the first sequence and bail out. Sometimes I read past you and you get me. Sometimes I read past both of you and she gets me.
Yeah, but is it the same? Will it be the same for the DNA? Is the same as the RNA, or it just indeed?
I'm not quite sure what you're asking here. Oh, you mean does it drop off at the same time? In other words,
does the same amount of termination sequences need to be the same for each one?
I think if this is what you're asking for, not necessarily. Let's suppose all 3 of your termination sequences in the same gene. And I'm the polymerase. I'm going to make some nucleotides. I'm going to make an RNA. Now, today, you get me and I drop off. But tomorrow, when we transcribe the same gene, I might just read past you and all the way past you, and then Sandy knocks me off. And then another time, maybe she'll get me out of there.
Does it depend on which gene it's reading?
It's a roll of dice.
Yeah, I think some genes have more termination sequences than others, but for an individual gene, it's a roll of the dice. There may be some genes like they may have 2 or 3 or something, others may have several. So I think there's probably some characteristics depending on just what the rest of the nucleotides sequences are and how tightly they bind to polymerase to see whether you're better off having a whole slew of terminators rather than just say 2 of them. But within that gene it's going to be pretty much random. Sometimes she kicks me off, sometimes you kick me up, sometimes she exiles me. Whoever. Can't tell from one day to the next who's going to get in here. Okay, so that's why in this case we had to read through the first termination sequence and partially through the second. We dropped off of the second and we never got to the third. So of course we're not going to have the third sequence in our precursor RNA. Okay, did that answer your question?
I think so.
Okay, good.
I may have to ask another question later.
Okay. All right. Well, far away.
No, I'm going to think about that.
Okay, all right.
Maybe I'll.
Okay, so what we've got here, folks, like I say, this is not an uncommon test question. This are some variations of, sometimes I do that. Draw a typical eukaryotic gene and mention all the sequences and what they do, and then the initial messenger, the initial RNA, and describe all that, and then the final messenger RNA. I sometimes do that. That's usually a 15-pointer, by the way, if I do all 3 of them. But, I may or may not have. I got the right to think of. Okay, but that's it.
Let's put that on the test. I think that's a good question.
Oh, it might be. Generally, you're going to have at least some part of that on the test. Okay. All right. Now,
I have a question.
yes?
What does the MG stand for the 7
Oh,
MG?
7-methylguanosine. What happens is you have guanosine, that's guanine and the sugar and the phosphate. And then at the number, guanine is a double ring, and the number 7 carbon, don't ask me what, I haven't taken organic for 25 years, but the number 7 carbon has a methyl group attached to it by an enzyme. And there is another kind of cap called a trimethylguanosine cap that certain RNAs, not messenger RNAs, have. and that one has methyl group put at 3 of the carbons of the guanosine. Okay, so that's what, and more on that later. That just, quick and dirty kind of. Okay, so RNA processing, we're looking at the polymerase II transcripts. In other words, the ones that are going to be coding, that make protein-coding genes. We're going to see 3 major events that have to happen. One of them, formation of what's called the 5' cap. And that's at 7-methylguanosine. Now, like I say, that is a tag saying I am a messenger RNA. Other messenger RNAs, or rather, other RNAs may well have 5' caps, but they're different, and the ribosome simply will not recognize a cap. Like I said, there's a certain class of RNAs that has a trimethylguanosine cap. Ribosomes will never read that. They'll never accidentally grab that and think it's codes for a protein because the cap is different. The ribosome will not recognize that cap. If you take a messenger RNA with enzymes, remove that cap, put it back into a cell or put in a test tube full of ribosomes, the ribosome doesn't know it's there. It's like an invisibility screen. Okay, so that's one thing. Second thing, adding that Polya Tail. Or what we call polyadenylation. Now the Polya Tail is unique, we'll say, pre-messenger RNA processing. The Polya Tail is unique to messenger RNAs. Only messenger RNAs have that. Now that actually gives the molecular biologist an easy go of things. Let's suppose for, say, a hybridization experiment, or microarrays, or something like that. We want to get total messenger RNA from cells, or tissue extract, or ground up embryo, whatever the case is. Okay, you grind up tissue, grind up cells, grind up embryos. Oh, no, don't do that. Okay, you do that kind of stuff, and you're going to get all kinds of RNAs. You're going to get ribosomal RNAs, tRNAs, messenger RNAs, whole bunch of regulatory RNAs that we'll mention later on in the class. You get RNAs up to, precursor RNAs in the nucleus, these nuclear RNAs, all kinds of RNAs that you don't want. But only messenger RNAs have the Polya Tail. So what you do is you take a chromatography column, those little porous beads, you percolate buffer through it. You buy these beads that have poly T on it. In other words, a whole bunch of thiones covalently attached to the bead. Well, A's and T's hydrogen bond together, so you pour your cell mesh down this column. Anything with a Polya Tail sticks to the column, anything without goes out the column into the sink, and you throw it into the sink and turn the water on or flush it down the toilet or whatever, and then you add some chemicals to separate the hydrogen bond, and what comes off the chemical, what comes off the column now is pure messenger RNA. Nice. Single shot purification of total messenger RNA for a mere $20 a pop. Well, probably about 40 now. Okay. So it's a simple one-step procedure. So that's really nice. That way you can extract messenger RNA, ignore all the other stuff, the other RNAs in the cell, and do your experiments. Okay, so that's a nice, neat little thing that relies on the fact that only messenger RNAs have the Polya Tail. Now last, but certainly not least, is a process known as RNA splicing. And splicing is where we remove the introns from that RNA precursor. We cut them out and discard them. And then we join the exons together. So it's like watching, taping something or making, a digital file of a network TV show and then putting it on your computer and then going with, one of those video editing programs and all the commercials, you just delete those sections and then you put the storyline sections together. So now you've got a new digital file that has a show minus all the commands for insurance, new cars, insurance companies, new drugs, all kinds of stuff. You throw that stuff out in the garbage where it belongs. Yes?
What is the last word?
Okay, together.
Together.
Okay. Now, as we're going to see, this RNA splicing actually is very important in regulation. You look puzzled. No? You look puzzled.
Tired but not puzzled.
All right. Be glad to see eyes red. Okay, so now for D month.
You gotta be careful, there are people coming.
Yeah, they'll probably come and shoot me in my office, right? Okay. Well, no, someone might be in business for themselves. No, it pays tuition, which is about 4 times what it was when I first came here. We actually had a grad student, in all seriousness, many years ago, a grad student who was doing some pharmaceutical manufacture without a license, and midway through his master's program, and it was already, it turned out we didn't know at the time, was already on probation from a previous drug dealing offense, so they threw his butt in jail and never came back. Yeah. Well, generally when you're on probation, means if you do this again, you will go back to jail and serve your original sentence, plus the second sentence we're going to toss down your throat too. So yeah, they put him away for a while, I guess. Okay, but anyway, okay. The splicing, as we're going to see, is not just cutting the intron sequences out. We can do things like deliberately discard exons or even parts of exons, what's called alternate splicing. And that explains why we have introns and exons to begin with. Because that way, you can make a whole family of proteins just by selecting what exons you're going to use and what exons you're going to toss out. So one cell type has one version of the protein, another cell type has a different version, a third cell type has still a different version by this alternate splicing, selecting what exons you keep and what exons you toss. And there's more complications too, we'll get to that later. But what that means is one single gene can code for a whole bunch of cell-specific versions of the same protein. Rather than having to have an individual gene for every single variant of that protein and every single cell type, you can do it all with one gene. Saves on DNA. And DNA is expensive, right? There are some cases, there's one particular gene that's estimated there are at least 30,000 possible combinations, whether they're all used or not, because it has so many different exons and introns. And different versions in a whole wide range of different cell types. Imagine if you had to code for 30,000 versions of the same protein in one genome. That's more than the total number of genes we have in our genome to begin with. And that's just one example. Dramatic one, admittedly, but that's alternate splicing we're going to see. Is splicing a very important way of regulating things at the level of RNA processing. Very important for regulation. So we're going to see that. Now here again, prokaryotes don't do that. Every single protein they make is coded for by one single gene. Sesame, for instance, we've got maybe 22,000 genes in our genome. It's estimated that we have 500,000, possibly as many as 1 million distinguishably different proteins in our bodies. Yet we're doing that on a 20th, in fact, on a 20th to a 50th of the number of genes that you would need to code for every single one of them individually. So that's pretty remarkable. Okay, well, that's what we see here. And this is, of course, for the messenger RNAs.
How many proteins did you say that we have?
I've heard estimations, it depends on what paper read, 400,000 to 1 million, somewhere in that general neck of the woods. So yeah, I guess they're making more like an order of magnitude estimate and stuff. And I've seen different numbers in different papers I've looked at. So it's a lot. It's a huge amount. So that's pretty interesting. Now, one or two things to mention. This is what we see in the case of the messenger RNA precursors. But other RNAs are also made as precursor molecules. In some cases, you're going to do the equivalent of splicing, where you're going to remove certain sequences and maybe glue some other sequences together. In other cases, you will chop a single RNA precursor into 2 or 3 pieces, and then use those pieces as the final RNAs. In still other cases, you may modify certain bases, you may make some kind of cap, any of a number of things. So in a lot of times, some kind of splicing or cutting, in other words, cutting a part off without gluing it to something, that's common for the other types of RNAs. The tRNA precursors, the ribosome RNA precursors, and so on. Probably, and many of these regulatory RNAs are seen to also be transcribed as larger precursors. So this idea of extensive RNA processing is a very, very important kind of thing. Yes?
I have a question. Okay, in like our genes we have exons and introns. Can an intron ever be part of like a different type of gene? I'm not a gene, but a different type of protein?
Well, as a matter of fact, it's not common, but there are sometimes protein coding genes within the introns. And there are also sometimes where you have, here's a gene, okay? And you make one kind of messenger RNA or RNA precursor from that. Sometimes you might also have, this is not too common, but you may also have a gene that effectively overlaps. You have a second promoter buried in here, and you'll basically have a second gene overlapping. That's part of the sequence of the first one. It has different signals for exons, introns, and stuff like that. So occasionally, a single stretch of DNA may actually include the instructions to make 2 different RNA precursors. That's not too common. It's more common actually in bacteria and stuff where you have a very compact genome, but there have been a few examples seen in eukaryotes. And the other thing, like I said, you can have genes in introns or partially in introns and overlapping with an exon. And those are protein coding genes. We're also finding regulatory RNA genes are sometimes located in or at least partially in introns. So yeah, it's a very, very good question. And it happens. Okay, that's kind of pretty neat. And especially now that we're seeing more and more regulatory RNAs, they find an interesting kind of thing when you looked at genomic sequences from a wide range of organisms. And look at the sequences that are most highly conserved. In other words, that change the least from even widely divergent organisms, like say roundworms in humans. Some of the most highly conserved sequences are not part of genes. Sometimes you find some in certain introns. Sometimes you find some between genes. Those sequences apparently, at least in some cases, are coding for critically important regulatory RNAs. So you find these kinds of things that are quite unusual. So sometimes they used to call the introns and all that stuff between genes, quote, junk DNA, like it's there, it's garbage, it's just genetic baggage in the stuff. Sometimes you get things like duplications of stuff and translocation stuff, and it just all piles up. It's kind of like Windows. You ever run Windows for like weeks on end? It eventually piles up with all kinds of little electronic garbage and finally slows the whole thing down, and you have no choice but reboot the computer. The older versions of Windows were especially like that. Windows is kind of notorious for piling up the garbage until it finally slows down and stuff. The newer version, running my one in the office for weeks on end, my one at home usually a week or two before I have to reboot. But the earlier versions were really good at piling up the garbage and finally got to the point of slowing things down. So that's what they used to think, these sequences that had nothing to do with genes were. Now we're realizing they have a lot of importance. It's just, for the most part, we don't know what that importance is. So when we start looking at regulated gene expression, there's so much cool stuff that's relatively recent. It's hopefully I've conveyed a little bit of that as we go through here. But anyway, a lot of the other RNAs you do have cutting things out, modifications, making caps in some cases, modification of certain bases, sometimes splicing, sometimes just cutting pieces out and tossing them in the trash and so on. Lots of regulation. Okay. Okay, I'm going to mention, because I can't quite get to the rest of this, I'm going to mention one thing here. Okay, an important thing. Now, originally when they isolated nuclei and they broke them from the rest of the cell, and they broke nuclei open and did all kinds of experiments, crude experiments to get the size distribution of things like RNAs and things like that. They found a very diverse distribution. All kinds of sizes from very small to thousands, even tens of thousands of nucleotides long. So they say the population was quite diverse, and one word for diverse, a fancy word for diverse, is heterogeneous. Heterogeneous, or heterogeneous. That means it's very diverse. So when they noticed that huge diversity of RNA, most of it they didn't know what the stuff was, and it was found in the nucleus, they started calling heterogeneous nuclear RNA, or hnRNA. You'll still see that term. That's the nuclear RNA. Some people just call it plain nuclear RNA. That's RNA found in the nucleus. Now, that's just found in the nucleus only. In the cytoplasm, you see plenty of RNAs, many of the messenger RNAs, tRNAs, of course, ribosomal RNAs, parts of the ribosome, other kinds of RNAs. But you don't see all this kind of mess that you have here, a huge distribution of various sizes and what have you in that. Now, one other thing we see, though, most of that stuff never leaves the nucleus. And if it stays around the nucleus long enough, it eventually gets degraded by nucleus. But what we're seeing when we look at things like RNA precursors, like messenger RNA precursors and stuff like that, those stay in the nucleus until they are fully and completely processed. If a messenger RNA precursor does not have that cap, the Polya Tail, the splicing is not happening, any or all those things, it will be stuck in the nucleus forever. Well, not forever. It will be stuck in the nucleus and then destroyed by nucleases. And as we're going to see, a lot of RNAs, a lot of precursor RNAs are made in the nucleus and they never get out alive. Sounds wasteful, but there's some reasons for it. You know, like they used to say in the Bible, many are called, but few are chosen. That applies to RNAs in the nucleus. Many of them are never chosen to go out in the cytoplasm because they're not properly processed. So already we have an important thing of regulation. We can decide whether or not to process a given RNA. If we process it, eventually, at least if it belongs to the cytoplasm, it'll go out in the cytoplasm. If we say, no, we are not going to process you, you're stuck in the nucleus until the day you die. It's probably not going to be too long from now. There's lots of hungry nucleuses floating around. So, that in itself, to process or not to process, that is the question. That in itself is an important means of regulation. So that's what we're going to look at when we come back on Thursday. Of course, we'll see you guys in the lab. And any questions in our last moment or two?